Hello, thank you for joining. My name is Aviv Mezer, and I'm from the Hebrew University in Jerusalem. I want to start and say thank you for the invitation to present my work here. And I'm going to talk today about using quantitative MRI to study the brain, in particular in aging and neurodegenerative diseases. When I got the invitation to present this work, I was looking backward on a quantitative MRI, and I remember my first visit to OHBM about 15 years ago. And I remember how small was the field, the quantitative MRI field back then. And it was great to think about the progress that we made. And I will start with my sense of a paradigm shift. I think the way we use MRI originally to research brain structure was using it as a camera. And we were looking on macro scale changes. We were looking on change in morphometry, in volume, and we were able to measure some change in the macro scale of the brain structure. And also clinicians were using the MRI in the same way, and also looking on bright or dark objects in the brain and relate them to clinical cases. Now, qMRI provide us a new input because now the images has units. So it's a physical measurement of the underlying tissue, for instance, of relaxation. And the analogy will be here a thermometer. And so now we can actually measure the body temperature with units and say there is a change in, say, 3 degrees in body temperature that relate to some disease, and not just to say someone is hot, right? So in the same way, we don't want to say a brain region is smaller or brighter. And in that sense, qMRI provides us a new way of measurement. And the next step was building models and relate the quantitative measurement to the underlying tissue, the underlying biology. And this idea is termed in vivo histology by Weisskopf and others. And the idea is that we try to relate the underlying tissue to the quantitative measurements. And this is where we are. And I think the future is us starting to look on the complexity of the biology. And of course, biology is a very complicated thing. And when we think about a very complicated environment, we need to combine multiple sources of quantitative measurement in a sophisticated way in order to sense the rich biology environment. And so I want to discuss that in particular in today's talk. So I say histology and how rich histology can be and tell us about the brain. And I want to share here a few examples. For instance, we know that we can look on lipid composition, and the lipid composition can vary as function of age. And if we look on lipid composition in the brain as function of age, we are able to predict the age in a very accurate way. If we look on cellular organization in space, we will find that it also varies in space, both in the cortex and in the white matter and in other regions, and that can allow us to identify functional regions, structural and functional regions, and also the molecular milieu of the tissue. That's something that we can measure with histology. For instance, we can look on iron, and change in the iron environment is actually indicative of different brain disorders. So there is a rich information that we know to measure with histology, and the need is to bring it to in vivo. Now, today I want to share with you 3 projects. One which we call tissue relaxivity and is sensitive to the lipid composition. The other one we term iron relaxivity and is sensitive to the iron environment. And the third, the special organization of the striatum. Let's start and look on the quantitative measurement. We see here a subset of measurement, and each of them is on a contrast because they are sensitive to different aspects of the environment, the biology environment, and they have different units. And we can assume that there are some function of the structural organization, iron and myelin. And the question, of course, is how to separate those things. Another important thing is to remember always is that we measure water. We measure water in MRI, and because we measure water, all our measurements are sensitive to the water fraction and environment. So let's start with myelin. And indeed, about 10 years ago, there was very interesting work that was comparing a T1, ex vivo T1 measurement to myelin staining. And T1 you can also use as 1 over T1, which is R1. And they find that about 90% of the variability in T1 in the white matter can be related to myelin. So this is great and important because it suggests to us that we have a measurement that is sensitive to myelin in a very strong way. And indeed, in the same year, we were doing this cross-section work where we measure R1 across the white matter, across different white matter pathways, and that was across ages. And we were finding that R1 is varying as function of age, and it looked like a parabola. There is an increase in R1 and then a decrease. And the rate of change is actually vary between different white matter pathways. Here the color code represents the rate of change in different white matter pathways in development and aging. And so we have here a measurement that is sensitive to the change in the white matter that can be compared across subject. And this work was replicated several times since. Now, the question is whether we actually measure myelin. And there are several quantitative measurements that's supposed to be sensitive to myelin. And so at around 2020, 2021, there was several reviews to try to answer this question, and they review multiple studies. And here is a summary of the result in one of them. In the x-axis, we have different quantitative measurements, different parameters, and the x-axis is the R-square, how much we can explain in terms of myelinization. And what we can see here is that there is variability between the different quantitative measurement, but also within the quantitative measurement. It's not one-to-one. Here is another one, and it's about 0.5 again for the different quantitative measurement, and even a third one, relatively the same result. Different quantitative measurement and the coefficient of determination, and the agreement is good, but it's not one-to-one. So if to summarize that, I will say it again. It seems that quantitative measurement in one hand are very sensitive to myelin, and on the other hand, they are not just a measurement of myelin. There is something else. They are not one-to-one with myelinization. That brings me to my first study that I want to share with you, and that will be about tissue relaxivity and lipid composition. So let's start with that. All the quantitative measurements supposed to be very sensitive to myelin concentration. So assuming we have a change in, say, a relaxation rate, and we have two measurements, one is higher than the other, we would assume that the concentration here of the myelin is higher. So if we will put here an x-axis of concentration, we assume that in this one there is more myelin and then the concentration is higher. But the question is, can we also imagine a case in which the concentration is the same, but the relaxation change? And from this review, it seems that we might. And if this is the case, it's a problem, but there is also information in it that we can try to retrieve and maybe say something about the environment, the molecular environment, which is not about the concentration. And maybe it will be about the lipid composition. So why do I say that? Let's first think about what is myelin. And when we look on a typical membrane, we would see that it's actually a very complicated structure with different type of phospholipid and protein inside it. So it's a complicated structure. And the question, do these different phospholipids can affect our signal in MRI? And the answer is yes. And this is actually a very old news. This is a work from about 30 years ago. Measure different type of phospholipid. Each of these rows is a different type of phospholipid, very common phospholipid, biological phospholipid in membrane like myelin. And the only thing they did is they changed the rate of the mixture. They didn't change the concentration. And the measurement show us that both T1, T2, and MT will change because of the molecular composition. So now, when we have a measurement of R1 and there is a change, say because of age, there is a question, is it about tissue function, something changed in terms of myelin concentration, or maybe it's about the tissue environment, something changed in the lipid composition, for instance. How can we answer that? There is ambiguity that we want to try to solve. So to do that, I will share with you a biophysical model that tries to assess the molecular change. I will validate it with in vitro experiment and then move to the in vivo application, and last, we will test it against ex vivo data. And all of this work is part of the PhD of Shir Filu, which is a very talented graduate student. Okay, so let's start with the biophysical model. We had these two measurements. They have a different relaxation, but the same concentration. So to understand that, we need a new term, and this term I will call relaxivity. And relaxivity is the ability of certain compound to increase the relaxation rate of the surrounding water proton spins. So it's how much a material can actually generate relaxation, and different material might have a different ability to do that, different relaxivity. And so if we'll measure them in different concentration, if we have 2 points, we can actually fit a line, and the line will be different for the different environment, different type of molecules. So we can put it in a simple equation, say R1 is a function of the concentration with some linear coefficient, which will be the relaxivity, the ability of the material to create relaxation. And this is great. This is actually explain the effect of the molecules. But the problem is that we can only measure that if we know the concentration. And so to know the concentration, we have to work either in ex vivo experiment or in vitro experiment. So the next question is, can we also do it in vivo? Can we find a way to do it in vivo? And here is our suggestions. We actually suggest that instead of using concentration, we use something else, and we would use the tissue fraction. What is that? So I will term tissue fraction as a measurement that's coming from water content. Water content is a measurement. It's a qMRI measurement that we can do in vivo, and it's coming from proton density. And if we do 1 minus water content, we get the lipid and macromolecule tissue fraction, MTV. And so it's a quantitative measurement with units, fraction of volume per voxel. And we can actually validate it. It's actually measuring the fraction of water to non-water. That's what we see here. And now, when we have this measurement, we can actually replace here concentration with MTV. And now we'll define our relaxation rate, say L1, as a function of MTV with some relaxation or some relaxivity. And this relaxivity will be the tissue relaxivity, which is the dependency of relaxation rate on tissue fraction. And the reason it's important and interesting is that we now have measurement that we can establish with in vivo, because MTV is something we can measure in vivo, and R1 we can measure in vivo. Okay, so let's first validate it in vitro. Okay, so we have R1 in here and MTV of 2 compounds, and we want to measure it with different concentration and see the different relaxivity. So that was exactly the work of a PhD student, Oshrat Stangel, which measure here different concentration of 2 in this image, 2 type of phospholipid, phosphatidylcholine and sphingomyelin, which are both very common in the brain and in tissue in general. Different dot, they are different concentration, MTV change, and we can see that the relaxation is different. Each type of phospholipid have its own relaxivity, different slope. Wonderful. So now let's try to do it in vivo. But note that we actually need to measure several MTV measurement. And for that, there is a question how we'll do that in vivo. So in vivo, in each voxel, we have only 1 MTV and 1 R1. How can we fit a line here? And the answer was, maybe we can use an RI approach. Now we'll have many voxels with many MTVs and many R1s. So here it is a distribution along the thalamus, and now we can fit a line and measure the relaxivity of the RI. So in phantoms, we saw that different phospholipid had different slope, had a different relaxivity. And also in the tissue, we can look on different RIs and see if they have different relaxivity. Here is the pallidum and a white matter RI, and you can see they have different slope. And we can generalize it to more RIs. Here we see 3 of them, and I'm going to show you many more. And I'm also going to do it on many subjects, not only on a single subject. So now we have several subjects, and this is the relaxivity on the different ROIs. And you can see that the relaxivity vary between ROIs, and that the variability between regions is smaller than the variability within subject. So we have a measurement of the relaxivity within an ROI. Now we don't have to do it with R1. We can actually replace the quantitative measurement in here. For instance, we can use MT, and we also get a linear dependency, and we also find relaxivity, and this relaxivity is different actually of this one. So there is a point to combine them. So now we can generalize that, and we can measure many relaxivity. In this case, I will suggest 4. There is the R1, R2, MT, and mid diffusivity, each of them. And we can measure the relaxivity of each of them across many brain regions, and that would get us a spider plot. And this spider plot is a signature of the region across different brain regions. And the shade here represents the variability across subject. So we have signature of brain regions of multiple relaxivities. Great. So we have a relaxivity in vivo on different RI. Can we relate it to ex vivo? So for that, we use a post-mortem tissue, and we did on this tissue both MRI and lipodomics and compare between them. So now, in lipodomics, we have many different types of phospholipid, and also in relaxivity, we have several measurements. So how would we do the comparison? We use PCA, and we compare the first principal component of lipodomics to the first principal component of tissue relaxivity, and that's what we see here. And we find agreement between them. We find that the relaxivity captured the change in the lipid composition, and we wouldn't be able to find it if we would use the quantitative measurement themselves. That's what we see in here. If we just use the quantitative measurement, they wouldn't agree with the lipid composition. Great. So we complete a circle, and we can ask, can we use it? And I will show the case of aging. So now, when we have a measurement, say a change here in an RI of the thalamus as function of age, there is a reduction in R1. And we can ask where it's coming from, and in this case, we can see that there is a change both in the molecular composition, so the relaxivity change, and in tissue fraction. There is a reduction in that. So in this case, both of them change, and we can look in more details on the tissue relaxivity and see that the one that changed was the MT relaxivity and the R1 relaxivity. This is the difference between the young group and the older group. But there are cases that only one of them changed. For instance, in this case, we see a change in R1, and this is due to a change in the relaxivity, so something in the molecular composition probably, but not in the tissue fraction. So that's also an option, and we can also see the other option when only the fraction change and not the relaxivity. So in general, it adds more information about the changes that we measure with quantitative MRI. Now, until now, I suggest that we can measure relaxivity on RI base, which limits our resolution. And the question, can we do that also in the voxel base level? And so to do that, I will share with you new work, unpublished work. And in this work, we try to ask ourselves, can we measure tissue relaxivity on the voxel level? And in order to measure it on the voxel level, we have a problem, right? We need to change the water fraction within the voxel. So, because we need at least 2 measurements to fit this line. But the problem is that tissue fraction is inherent property of the tissue. So we cannot change it. So we thought that maybe we can do an apparent experiment, and we're going to use MT. And what MT is doing is silencing part of this water. This is the water that is affected by the magnetization transfer. So some of the water are silent. And so we have the saturated water, which are silent, and the unsaturated water, which are still visible. And so now we have 2 conditions. One condition in which we see all of the water, and in the other one we see only the unsaturated water. So we can think about an experiment in which we measure R1 and tissue fraction once without MT. We see all the voxel, all the water in the voxel. And in the other one, we do a saturation and we see only part of the water. So we have a different tissue fraction and different R1. Now we have 2 points along this line per voxel, and we can fit a line. So we can fit a line, and this is an example for an ROI. And so we see here for each voxel we see the unsaturated values and the saturated values, and we can see we can fit a line. And you can see that this will be the line if we will fit across the ROI. And we can see also that now if we do it per voxel, we find something along the same values. Actually, the average of that is the average of those lines. So we got essentially relaxivity per voxel. And now we have a map. We have tissue relaxivity map, a new quantitative measurement in the brain. Okay. One more thing that we can do when we have a model, when we have the equation, is actually play with this equation and do these experimental thoughts, in which we will change the tissue fraction and try to imagine or predict what will be the R1. So what we see here is a movie in which we're moving the tissue fraction, and you can see the contrast is actually varying. It's starting with something which is actually flip, and reminds us something of development when there is tissue fraction is very low. And as we increase the tissue fraction, we're getting to something that looks like R1. And I think this is this experiment, or this thought experiment, can be very useful when thinking of dynamic change in the brain, for instance, along development and aging. Okay, so to summarize this part, I was talking about tissue relaxivity. I was showing that in lipid samples, we can measure tissue relaxivity, and it actually sample the molecular environment. In the brain, we saw that we can measure tissue relaxivity, and when we do many of those, we get a unique signature for different brain regions. And this multidimensional relaxivity was sensitive to the molecular composition that we measure with lipodomics. And in that sense, it allows us a better understanding of changes that we measure, say, with aging. And last, I show you the difference between ROI-based analysis and voxel-based tissue relaxivity that we're now developing. Okay, so that was tissue relaxivity, and now I want to talk about iron relaxivity. So here again, we have subset of measurements which are sensitive to iron. We know they're sensitive to iron, and iron is a very important component of the brain, and it's highly regulated. And the homeostasis of iron is really regulated, and if it changes, it actually have a very strong manifestation on the tissue. There are two main forms of iron that we can find in the tissue. There is the transferrin, iron within transferrin, which is the protein that transfers iron to the tissue, and there is iron within ferritin, which is the protein that aggregate and store iron in the tissue. Now, any change in iron could be part of different conditions such as aging, neurodegenerative disease, and cancer. I said before, we know that quantitative measurements are sensitive to iron. For instance, it's really known that R1 and QSM can be used as a proxy for iron concentration. And in here we see R2 star against iron is measured in ex vivo across different brain regions and also across ages, and we find a good agreement between them. Now, the problem is that it's really hard to measure the iron milieu, the forms in which iron is in vivo, and so this is a need. And I would like to suggest a way to measure that or sense that, and for that we will use two quantitative measurements which are sensitive to iron, both R2 star and R1, and we use the fact that each of them is sensitive to iron in a very different way. R2 star is very sensitive to the mesoscopic effects of iron, while R1 is sensitive to iron but in a local way. So it's a local interaction between iron and water that change R1. So we have here two different mechanisms. Again, it's a work of Shih-Filu, still unpublished. Okay, so we start with the biophysical model. So here is the idea. Similar to what we had before, we have here relaxation rate, and we have iron concentration. And if we have different iron forms, then they would have different relaxivity, which we can only measure ex vivo or in vitro. And that could be different relaxation rate. That could be either R1 or R2 star. So both are depending on the iron concentration, but with different relaxivity. Now, we cannot measure that in vivo because we need to know the iron concentration, but we can measure R1 and R2 star in vivo. So the idea was to maybe rearrange this equation and now plot R1 against R2 star. And also this one will be linear, and it will look like something like that. So R1 will be a linear function of R2 star, and the parameter here will be the ratio of the relaxivities of the iron relaxivity that we can measure ex vivo or in vitro, but this one can be measured in vivo. So let's see that using an in vitro experiment. This is an experiment that Ron Sharabani, a postdoc in my lab, did. And here we see different iron forms, iron with different environment, with ferritin and transferrin, with and without lipids. And different dots here are different concentration. X-axis is R2 star, Y-axis is R1. And we can see as we change the concentration, the relaxation, both relaxation change, but they're doing it with different slope. So the slope here of R1 to R2 star is indicative of the iron environment. And here we can see the R1 to R2 star relaxivity. And we actually have a prediction for that. The prediction was that will be the ratio of the relaxivity that we can measure in vitro. And since this was done in vitro, we can actually check, and the prediction is very good. You can see here the red dot that goes with the R1 to R2 star relaxivity. So now let's go to in vivo experiment, and we can use the RI approach, the same idea. Here's a single subject, and we see here 4 different RI, R1 against R2 star. And again, we see linear dependency, and the linear dependency is different for different ROIs. Can we relate that to histology? So before we said that R2 star is very sensitive to the iron concentration. R1 to R2 star is not sensitive to the iron concentration, but it is sensitive to the iron mobilization. This is a measure that can be done ex vivo, and it's a measurement of the iron environment or the iron homeostasis. So here again, we have a new measurement of relaxivity, which is sensitive to the molecular environment of iron versus the concentration. We actually wanted to validate that in the same subject, and so we collaborate with Tal Shachar. We measure patients which have a meningioma, which is a tumor. We measure quantitative measurement before their surgery. Then the tumor were resected and been sent to histology, so we can compare the iron homeostasis to the measurement of relaxivity. And to summarize, what we saw there is that we can measure, we estimate the iron mobility by the ratio of transferrin to ferritin, and we find that it's actually, if we separate into 2 groups, the one which have high iron mobility and low iron mobility, they goes with the R1 to R2 star relaxivity. So R1 to R2 star relaxivity was sensitive to the iron mobility, which is something we couldn't do if we would use just R1 or just R2 star. So again, we're having new information about iron mobility using the relaxivity. So to summarize this part, we first did use a model and checked it with in vitro experiment and find that R1 to R2 star relaxivity is different for different iron forms. We then saw it in vivo in the brain, and we found that the R1 to R2 star relaxivity is changing or varying in different brain regions. We showed that the iron homeostasis, which is a measurement that we can measure ex vivo, agreed with the R1 to R2 star relaxivity, and we saw it in across brain regions and in the case of tumors. And in general, what we have here is a way to enhance the specificity of MRI to the molecular iron environment. Great. So we saw the tissue relaxivity, which were related to lipid composition, and iron relaxivity, which is sensitive to the iron environment. And now I want to talk about spatial organization. I'm going to focus on the striatum. So the striatum is built from the putamen and the caudae, and this is the input structure of the basal ganglia. Here it is, and it's part of multiple circuits in the brain, cognitive and motor circuits, and it's also very known in the different brain conditions and symptoms, for instance, in movement and cognitive disorders, and maybe the most known is Parkinson's disease. So let's look on the striatum, and when we look on it with histology, we discover that it's a really heterogeneous structure. Here we see early work that look on neurochemical gradients, which represent different type of cells along the striatum, and there is a different in space along both the putamen and the caudae. And this cellular change in space also goes with connectivity gradient. This is with tracing. And we can see that there is a gradient in connectivity of the striatum, and that in the end goes with a different function, the most posterior area related to sensor and motor regions, and then it moves to associative, and in the most anterior part to limbic circuitries. Now, in Parkinson, we actually know that this gradient is altered. And if we do a measurement like PET in PD Parkinson patient versus normal, we will find that there is variability in the dopamine along the striatum. So here we see a different type of PET measurement that is sensitive to dopamine in the tissue. And in general, we will see that the most posterior aspect is changing in the patient, but not in the control. And so there is effect in the gradient in the posterior area, and this change is typically also asymmetric. The disease is asymmetric, and so there is one side which is affected more than the other. Okay, so I want to suggest a tool to identify this striatal gradient, and then we'll use multiple quantitative measurement to measure this gradient in control and in patient, and then compare it to the structure. I think it's important to relate our quantitative measurement to behavior, so that we relate it to symptoms. And last, we also generalize it to non-quantitative measurement, clinical data. Let's start with the tool. So this is a work of a very talented graduate student, Elior Drori, and he developed a tool to identify a striatal gradient. And the idea is as follows: we first identify the striatum, and then we use the coordinate themselves, and using SVD analysis to find the main axis of the coordinate of the striatum of the specific subject. In this case, this is the putamen. So now when we have the main axis, and I'm here looking on one axis, the anterior posterior, I'm going to focus on it today. And we subdivide it to different subregions, and we sample quantitative value along it. For instance, R1 here on a single subject, and we see the change in R1 along the putamen. Now, the importance of this type of analysis is that we didn't need to align all the brain to the same space. We did it on the ROI coordinate, and we didn't have to also respect the coordinate of the whole brain. It was something about the putamen of this subject. And we can compare it across different subjects. So here is a comparison across different subjects, from anterior to posterior, and you can see that all of them show a gradient in R1. Now, when we have a very consistent measurement across subject, we can actually define the norm. So we have a norm of variability in R1 in space, and we can compare it now between different groups, for instance, between patient and control. So that's what we're going to do now, and with multiple quantitative measurement. So in collaboration with Gilad Yalom, we measure a patient and healthy controls. We measure multiple quantitative measurement and different clinical measurement on the patients and compare all of those. So in a work that is still unpublished, we measure the quantitative measurement on these patients. We identify the main axes. Notice that those axes is something that we're doing per subject. So we can sample now all the quantitative measurement on the same axis. And now when we do that, we can find that each of these quantitative measurement have a gradient. That's what we see here. And that in some of them, we find a change between a healthy control and a Parkinson patient. And this change is in particular in the posterior aspect of the putamen. The biggest effect here is with MTV, the fraction of water to non-water. That goes with atrophy. Now, what we can do next is try to relate this change to the symptoms. So the main symptom in Parkinson is motor symptoms, and we already said that they are asymmetric. There's a difference between the right and left. So we can try to relate the asymmetry in motor symptoms to the asymmetry in the quantitative measurement. So in one hand it will be the non-water fraction, and in the other hand will be the motor symptom asymmetry, which is a clinical assessment. And you can see that we find an agreement or correlation between the MTV measurement and the motor symptoms. And if we do the same analysis to AutoStar, which has also been suggested to be a measure for Parkinson, in the same posterior aspect, we actually couldn't find an agreement between them. So we can say that, at least in this data, the change that we find in microstructure that can be related to the motor symptoms coming from change in water fraction increase, which go with the atrophy. So now we ask ourselves, can we actually generalize it to clinical data? Because qMRI is wonderful, but most of the data that exist right now are not quantitative, particularly for patients. So here we compare to the PPMI data set, which have a lot of MRI in clinical setting. And in this clinical setting, instead of using quantitative measurement, we use the T1 weighted versus T2 weighted measurement. So this is the measurement that we use, which is a semi-quantitative measurement. It's not a quantitative measurement. So we ask first, does it have a gradient? So here's the gradient that we measure with T1 divided by T2 along the putamen using the same analysis. Now we can ask, is it similar to R1? And there is a very good agreement between a gradient in R1 and gradient of T1 divided by T2. This is great. Next, we can ask, do we find a difference between patient and control? And we do find a difference. We find interaction between position and group. So there is a change in the posterior aspect of the putamen also in this database. Now, in this database, there is also clinical data, so we can compare this measurement also to the clinical data. So comparing the asymmetry in the motor symptom to the asymmetry in the semi-quantitative measurement in the posterior putamen. And here again, we find agreement between the motor asymmetry and the structural measurement of the putamen, the posterior putamen. An interesting point about this data set is that we also have a PET and DAT scan, which is a measurement, direct measurement of dopamine of those patients. And so we can also compare the asymmetry in the structure in T1 divided by T2 to the DAT asymmetry, which is a measurement of the uptake of dopamine. And here we also find agreement. So the asymmetry that we measure with the MRI using this analysis actually agree with what we can measure with a more complicated DAT scan. And if you want to hear more about what we did with this data, we actually did now new analysis, you can come and talk with me. I will be near this post. So to summarize this part, I describe a new tool to identify a gradient along the striatum. I showed that we can sample multiple quantitative measurements along it and compare between patient and control of Parkinson, and we find difference between them. We find that this difference, in particular of MTV, can explain the variability in motor symptoms of subject, and we can also generalize that to clinical data with T1 divided by T2. To summarize, I started with the importance of histology. I said that with histology we can measure the molecular environment and get a very rich picture of the tissue. We talked about I mentioned lipid composition, I mentioned cellular organization, and iron homeostasis. I then said that, and using the quantitative MRI, we now at the level of measuring the concentration, and the future is creating new models that will allow us to measure the complexity of the tissue, combining multiple quantitative measurement. And then I showed you 3 examples of that. I showed you the tissue relaxivity, which is sensitive to molecular and lipid composition. I show you the iron relaxivity, which is sensitive to iron environment. And last, I show you the multi-quantitative measurement along the striatum, along the gradient of the striatum. And with that, I will end. I want to say thank you to my collaborator, Tal and Gilad. In particular, I want to say thank you to the student who did the work, Shir and Elio, Ostrat and Rona. I'm thanking the funding agency, and thank you for listening. Thank you.